Okay, we're set to go. Our next speaker is Cori Bergman, who is a neuroscientist, a professor at Rockefeller University. She was an undergrad at University of Georgia. She then did her graduate work at MIT with Bob Weinberg, where she worked on the molecular biology of cancer and oncogenes. She then did a postdoctoral fellowship with Bob Horvitz at MIT, where she did really molecular biology of neuroscience. And she discovered really that an interesting worm, the C. elegans, which she will explain to you, has the sense of smell and can detect really literally hundreds of different odors. Although it's a very simple nematode, a thread-like worm, which has only 302 neurons, a very simple system. She went from her postdoc to UCSF, where she made a number of remarkable discoveries and studied the complex behavior and such as feeding of this tiny little worm. She discovered the SIG-1 system, which is a matchmaker, a molecule that directs neurons, which was, again, a great discovery. In 2004, she moved to Rockefeller University, something to do with getting hooked up with Professor Richard Axel, who is her husband. And so she's been in New York City, really, for the last 10 years. And here she's going on with studying neuroscience of odor in the olfactory system, something, of course, that Richard has a Nobel Prize in elucidating in another system. With this C. elegans system that she's been working with, she's found some remarkable thing. For instance, if this worm goes and munches on bacteria that have a specific odor, and that bacteria makes the worm sick, that worm, the next time it sees a bacteria or smells a bacteria without odor, will move away from that bacteria. So there's an environment impacting the nervous system of a worm, which is quite remarkable. And so that's learned olfactory behavior, which is something that she has really concentrated on. And she has, for instance, another single gene in C. elegans determines whether the animal will prefer to eat alone or in a social group. That's remarkable. That is amazing. And so with all this, Cori has received many honors and many awards. Among them, she's a member of the National Academy of Sciences, the American Academy of Arts and Sciences. She's received the Kavli Prize, the Breakthrough Prize in Life Sciences. She co-chairs now the President Obama Brain Initiative, and that's a big deal with recognition right in the White House that neuroscience is moving very rapidly, and it's very important as potential background for discovery of new drugs, which are so needed in the areas of neurodegeneration. So I would like Cori to step up now. She's going to talk on fixed circuits and flexible behavior. Cori.
So I'd actually like to start this talk with a moment of appreciation in the context of the last session for Roy Vagelos in particular and Merck, and to point out that drug companies are not just for rich countries and rich people, and that through its development of nematicides, particular ivermectin and related drugs, Merck treated, I heard last night, 200 million people in the developing world for diseases like river blindness, free of charge, and that benefits that come from this industry really benefit the world and deserve all our appreciation. There is a tremendous unmet need for treatments for neurological and psychiatric disorders. About a third of all people will at some point in their life suffer from a severe disorder that impairs their quality of life, whether we're talking about the developmental disorders of children, Am I on? Such as autism, or the diseases that affect the cognition and emotion of young adults, such as schizophrenia, or the dementias and degenerative diseases that strike older adults. And we can do far too little for these people now. Now, in my talk today, I am not going to address that need. But what I am going to do is to try to talk about the biology of the systems that we do have, the brain systems that are targeted by the treatments we have today, something about how those work, and perhaps how that might give insights into developing future generations of treatments that interact with the brain. So neuroscientists as a whole are really interested in fast chemical transmission and fast electrical transmission of action potentials and transmitters like glutamate and GABA that transmit information in tens of milliseconds at synapses. But in parallel to these fast systems, neurons in the nervous system release small molecule neuromodulators like dopamine and serotonin, and neuropeptides, a class of small molecules of which there are about 100 different examples in the human brain, which also act on neurons at slower timescales and in more subtle ways. They represent, although they do not represent the dominant fast mode of transmission in the brain, they do represent the major targets of many of the successful drugs we have today to treat neurological and psychiatric disorders. So whether you're talking about L-DOPA in Parkinson's disease, or the atypical antipsychotics used in schizophrenia, or the mood stabilizers, all of which affect dopamine and serotonin, you're looking at drugs that affect these modulatory systems, the more subtle systems that act in the nervous system. And in addition, the many neuropeptides are very poorly studied, but they represent regulators of important physiological and neurological processes like sleep, hunger, pain, stress, and social affiliation. And all of these molecules signal primarily through G-protein coupled receptors. They're released sometimes at synapses or near synapses, but they can diffuse some distance away, in general not known exactly how much. In some cases, they're made deep within nuclei in the brainstem or hypothalamus, and then processes transmit this information to different parts of the brain. Understanding how they function further might help us to understand how they act. Now, in my lab, we study these systems not in the human brain with its 86 billion neurons, but rather in the very simple nervous system of the nematode worm Caenorhabditis elegans. Instead of billions of neurons, C. elegans has only 302. Instead of trillions of synapses, it has about 7,000. But at a genetic level, C. elegans has surprisingly almost as complex a repertoire of molecules functioning in its nervous system as function in the human nervous system, 20,000 genes instead of 25,000. And even more remarkably, most of the genes that we know about that, for example, have through recent studies been implicated in human neurological and psychiatric disorders represent old genes, genes that are present in recognizable form in the nervous system of the tiny worm as well as in humans. And in particular, I'd like to mention that there are about 100 neuropeptide genes in the worm, just as there are about 100 neuropeptide genes in the human. And some of those we can even recognizably detect a relationship between their function. So just to give you one example of that, perhaps the clearest case you could make for a molecule that's important in behavior in mammals is represented by the small molecule neuropeptides, oxytocin and vasopressin, two related molecules, each 9 amino acids long, that signal through related G-protein coupled receptors. And these molecules are important both in mammalian physiology and in mammalian behavior, and specifically in reproductive behavior. So when a female mammal gives birth, her body is flooded with oxytocin released from the hypothalamus through the pituitary that induces uterine labor. It induces the milk letdown response for lactation. And in addition, oxytocin released in the brain causes the female to generate a set of strong behaviors associated with maternal care, caring for the pups, maternal aggression, and so on. Oxytocin, therefore, really represents a very strong link between behavior and physiology of maternal care for offspring, which is what we do as mammals. But in addition to this role, oxytocin has roles in the brain of all mammals at all developmental stages, and vasopressin, the cousin peptide, also has related roles. in a series of different kinds of functions like social recognition, pair bonding, and mating. So a few years ago, we noticed that in the C. elegans genome, there was a molecule related to oxytocin and vasopressin, a neuropeptide encoded by a single gene. We also noticed that there were two receptors in the C. elegans genome that were G protein coupled receptors that were clearly more similar to oxytocin and vasopressin receptors than either was to any other gene in either of these organisms. So there seemed to be molecular similarities between this peptide and its receptors across species. This carries out as well to the level of biochemical similarity in the sense that the C. elegans oxytocin-related peptide interacts with the C. elegans oxytocin-related receptors with nanomolar affinity, suggesting that this relationship is authentic. But the really interesting thing about this molecule is not necessarily its biochemistry, but its function. And when Jennifer Garrison, a postdoc in the lab, looked at the function of the oxytocin-related system in C. elegans, she found that these molecules were required for reproductive behavior in this animal. That one of the neurons that makes oxytocin is prominently associated with structures in the tail of the male C. elegans that are required for mating. Many of the neurons that are involved in mating express receptors for this peptide, and animals that are defective in either the peptide or its receptors are defective in mating behaviors in what we consider an interesting way. They're capable of carrying out all the actions involved in mating, the motor actions, but they perform them in abnormal sequences and with abnormal timing. They can't organize the behavior over a long time. So this first conclusion that I want to make about neuropeptides is that they can have remarkably specific biological effects, sort of clear functional effects, unlike a molecule like glutamate, for example, which might be found in synapses throughout the brain and that you wouldn't think of as a behavioral molecule in the same way. And second, that some of those effects may actually be related to particular biological functions that you can recognize across evolutionary distance. So even though mammals and C. elegans are very different animals and that their reproductive behaviors have completely different levels of complexity, are controlled by different cues, represent different actions, there is at the core of both of them regulation by a set of recognizable peptides that are present across evolution. And work from Bill Kristen's lab at UCSD has indicated that in yet the third branch of vertebrate evolution, the Lophotrochozoa, which are very distant both from C. elegans and vertebrates, oxytocin-related peptides also play a role in reproduction, suggesting that these molecules really did get locked into behavior at a very early time in evolution. So conservation is striking, but at some level, what we really want to do when we work on C. elegans is to try to be able to address questions at a different level of detail than simply saying there is a gene and it affects a behavior. What we want to be able to do is to really fill in the intermediate zone between the level of molecules and synapses on the one hand and the level of the behavior of the entire organism on the other hand. So from work in mammalian systems, we know a great deal about how neuromodulators and neuropeptides function to activate G protein coupled receptors in their signaling pathways. We also know from pharmacology that these molecules can be potent regulators of motivational and emotional states. They're the targets of drugs of addiction, for example, as well as of psychiatric drugs. But somewhere between this very high resolution view and this large-term behavioral view is the brain and the neurons within the brain and their function and connections with each other. And the question that we can address in C. elegans that's very difficult to address in a much more complex brain is what the structure is of the neuromodulatory circuits. How the neuromodulators work, what kinds of distances and timescales they work over, and how they relate to the fast synaptic circuits that are best known for driving perception, action, and locomotion. And we can do that in the context of really a fantastic map of the nervous system, shown here in its entirety through the work of John White and Sidney Brenner and colleagues, where every single neuron in C. elegans was reconstructed from electron micrographs, forming a connectome, a set of synaptic connections between each of these neurons. And we can really look in every single worm, identify each neuron, know what other neurons it's connected to, know what genes it expresses, and know what its basic functions are. So for example, from experiments where individual neurons have been killed or inactivated, we can say for most of the neurons in the nervous system, it is required for this function or for that function. And this map then is what we want to use. We want to be able to superimpose the role of neuromodulators and neuropeptides on this synaptic map and on this functional map to understand how they regulate behavior. And I'm going to take you through a couple of examples of how we've studied these maps in C. elegans, looking at different kinds of behavior. And the first one I'm going to talk about is social behavior. But it's actually not going to be the mating behavior that I alluded to briefly, that's regulated by oxytocin homologs. It's a non-mating behavior, a social aggregation behavior. And this social aggregation behavior is regulated by stress, actually a common feature of aggregation or social interactions in many organisms. And it's also regulated by a neuropeptide receptor, a molecule called NPR1. So for every action, there is an equal and opposite reaction. If oxytocin is the social neuropeptide, then NPR1 is the antisocial neuropeptide receptor. High levels of activity of this particular neuropeptide receptor cause animals to have a solitary, a preferential solitary behavior, where they scatter across one another, apart from one another to feed. But low levels of activity of this same neuropeptide receptor, including the alleles that we see in most wild-caught strains of C. elegans, have a highly aggregating behavior and therefore gather together in these feeding groups. And the difference between these two behaviors is regulated both by environmental conditions and by this genetic variation. The stress levels can lead animals to very rapidly switch their behavior between these two behavioral strategies. And what the activity of the neuropeptide receptor does is to change the threshold for these stressful stimuli. So animals will switch between these behaviors at either relatively low levels or much higher levels of the stressors, depending on their allele of the neuropeptide receptor. So moving from a molecule, a G protein coupled neuropeptide receptor, to a circuit, we can first examine the expression pattern of this molecule. And what we find is that the neuropeptide receptor is expressed in about 10% of the neurons in the C. elegans nervous system. But we don't have to be limited by that expression pattern. We can narrow it down further to ask where it's actually required to carry out its functions. And Evan McCosker, when he was a graduate student in the lab, found that there was really a single pair of neurons, a pair of neurons called RMG, these guys back here, one on the left and one on the right, that was the essential site of action in regulating aggregation. That if the neuropeptide receptor was expressed just here, the animals showed low aggregation behavior. So we can take that map now and turn back to the wiring diagram. And looking at the wiring diagram gives us a very clear model immediately for how this behavior is generated. The RMG neuron that expresses the neuropeptide receptor is linked directly to synaptic connections to a variety of different signals that are important in aggregation. Two of the neurons that synapse directly with RMG detect pheromones made by other worms, which are among the cues the animals use to aggregate together. Two of the other neurons sense different things about stressful contexts, nociceptive contexts and oxygen levels, which then regulate these kinds of inputs. So you can see here a little synaptic motif that's holding together the pheromones, the stressful cues and contexts that trigger the behavior, and the site of neuropeptide action. And what the role is of NPR1 is effectively to silence this central hub neuron in the circuit and thereby to unlink the connections or to weaken the connections between these different context and pheromone cues. So at the level of a circuit, the sensory cues and the neuropeptide cues are directly integrated in the neurons that detect both. So here's one view of what a neuropeptide does in a behavior. It's effectively diminishing or damping down the circuit that drives a behavior, in this case, aggregation. But you can actually take this same circuit and sort of rearrange it the way you would rearrange a mathematical equation in terms of x instead of y, and get a different view of what's going on with respect to the neuropeptide receptors. So for example, instead of looking at this hub neuron here, let's look at one of the spokes, one of the sensory spokes that detects pheromone, this triangle neuron here called ADL. So the spoke neuron ADL detects C9 pheromones. We can see that, for example, using functional calcium imaging experiments. And it detects this pheromone in both social and solitary strains. It only drives attraction or aggregation in the social strain, the strain with low levels of activity of the neuropeptide receptor. But the solitary strain is not blind to this pheromone. In fact, the solitary strain is appalled by this pheromone and is strongly repelled from it. And this is part of how the animals scatter in a biological context. So Hyun Jang, a student in the lab, together with our collaborators from Piali Sengupta's lab at Brandeis, mapped out the nature of this repulsive behavior. The repulsive behavior is driven by the exact same sensory neuron, but it's driven by a different set of synapses downstream of the same sensory neuron. So this one sensory neuron is connected to the hub and spoke circuit that's regulated by neuropeptide receptor. It also forms chemical synapses onto a different set of downstream target neurons. And those different neurons drive an alternative behavior, actually an antagonistic behavior of repulsion compared to attraction. So if we view the role of the neuropeptide receptor from the point of view of pheromone detection, what we see is that this neuropeptide is toggling pheromone responses between two alternative behavioral outputs of attraction and repulsion, and that they're really antagonistic circuits to which a single input is attached that are regulated by the neuropeptide receptors. And we think this is an insight into the kinds of ways that neuropeptides may be functioning more generally. That what they are doing is helping circuits to drive the choice between alternative behavioral outputs by preferentially strengthening or weakening the different outputs. And when we look at the nervous system of C. elegans, and even more when we look at the nervous system of more complex animals, we see that in fact there are many different kinds of outputs from any given input, that neural pathways bifurcate, that C. elegans neurons might connect to 20 other neurons, mammalian neurons might connect to 1,000 other neurons, and that these could represent really different kinds of outputs that can be shaped by these other kinds of signals. So if we think of wiring diagrams as including different classes of synapses, we can include not only the different classes of fast synapses, like chemical synapses and gap junctions, we can include these slower synapses as well, the neuropeptides and biogenic amines. But we have a problem here because these are not recognizable in electron micrographs. We have to be able to map them out because they act extrasynaptically using other strategies. So our wiring diagram is both ambiguous because there are multiple potential outputs and incomplete because it lacks these modulatory signals. So how can we study those? What can we learn about those? So to tell you a little bit more about how we figure out a wiring diagram and really map the modulatory circuit, I'm going to move to another behavior that we look at as a model of a different kind of motivated behavior of C. elegans, and that's a foraging behavior. It's actually a very simple foraging behavior. It represents two different arousal states of the C. elegans. One state is kind of a quiet state in which it's eating, moving slowly, laying eggs, and that's called dwelling. And then its other state is a very high activity state in which it's moving around very actively, covering a broad area, but also still eating and laying eggs and so forth. And these two different foraging states are long-lasting. They're regulated by internal states as well as external cues, but it appears that the animal unpredictably transitions between them. It seems to make a decision by itself as to when it's going to do one and do the other. And Steve Flavell, a postdoc in the lab, has analyzed these behaviors very carefully and quantitatively. And that involves tracking animals for long periods of time and looking at things like what patterns of behavior are represented in each of these states, and how long do they last, and can we see any patterns in when animals switch between them. And the main point I want to make about this is that you can really analyze a behavior quantitatively in terms of different kinds of motor actions. But if you looked at the animal for just a second, or if you looked at any one action, it could be doing that either while roaming or by dwelling. It's a large pattern of these actions that really distinguish these two behavioral states. And we can categorize them. And one of the interesting things about them is they both last for a long time. This is a 90-minute movie, and what you can see is that here for about 30 minutes in the middle, the animal was engaged in a behavior with high speed here and low turning rates, a 30-minute long roam, and then that was followed by a 30-minute long dwell. And on average, these states survive for about 8 or 9 minutes. So one of the things that we're curious about is what is it that would generate this kind of long time scale in the nervous system? We think these long time scales are interesting because we think they might serve as models more generally for things like motivational and emotional states. We don't think this represents any particular emotional state that you might have, just that it's a representation of these kinds of long-lasting states that arise in the nervous system. So Steve did a screen for molecules that affected these behaviors. And what he found is that neuromodulators are really important in these behaviors as they are in the social behaviors I told you about earlier. In particular, the small molecule modulator serotonin is essential for normal dwelling behavior. And what happens in serotonin defective mutants is that dwelling states are much shorter than normal, just a few minutes long, and roaming states are much longer than normal. And conversely, Steve identified a neuropeptide that is required for normal roaming behavior, that animals that lack this neuropeptide will sometimes dwell for hours at a time. And here again, roaming states and dwelling states, when they occur, are normal, but the dwelling states are much longer and the roaming states are much shorter when animals are defective in this peptide. So again, our real question here was to try to understand what the circuits are that give rise to these behaviors. And to study those circuits, we can again map what these different genes are, what the molecules that make the modulators, and what the molecules are that make the receptors. We know, for example, that in C. elegans there are exactly 11 neurons that make serotonin. Now I should mention that serotonin affects many C. elegans behaviors, including egg laying and locomotion and feeding. It has multiple receptors. And the behaviors that we want to look at are not just serotonin in general, but really very selectively, what is it that's regulating these long-lasting foraging states? And so Steve addressed that by using intersectional Cri-Lox type methods to alter serotonin or its receptors in different cell types. And he found that a specific subset of serotonin expressing neurons and a specific subset of serotonin responsive neurons regulate the roaming and dwelling transitions. And I just really kind of want to bring this point home perhaps in this audience. So there are many drugs out there to treat mood disorders, anxiety disorders that target the serotonin system now. Now, it tends to target the system as a whole. So when we look at the serotonin system for roaming and dwelling, we find that some of the serotonin neurons, but not others, are important. We find that one of the serotonin receptors, but not others, is important. And in the pharmacological industry, of course, the level of the receptor is usually the level at which targeting takes place when looking at when trying to develop functions. But we also find that a subset of the places that the receptor expressed are important, and another subset are not. So it's not serotonin as a whole that's important, it's serotonin from certain sources and serotonin on certain targets, as well as a particular serotonin receptor that are important. And I think in terms of possible ways of thinking about sort of the next generation of pharmacology for brain disorders, trying to think about these more localized functions instead of very general functions might be an avenue to pursue. The same kinds of studies of the neuropeptide system lead to very similar conclusions. So again, there are multiple neurons that make the neuropeptide that's required for roaming, and there are multiple neurons that must express its receptor, but these represent a subset of the neurons that make the peptide and a subset of the neurons that express the receptor that are really tied into this behavior. We can now take these molecules and take this wiring diagram here that we've drawn in red and in green that represents the modulatory network for serotonin and PDF, and ask how that relates to the wiring diagram of the worm. And what we found was really something of a shock, which is that these neurons are completely unconnected to one another by synapses. None of these neurons that are linked by red or green arrows are linked to one another by classical fast synapses. So this represents a wireless, in a sense, that you cannot recognize it at all through electron micrographs. It has to be solved separately. So in addition to asking which neurons make these, we can also ask when those neurons function to carry out these behaviors. We can do that by using methods to directly monitor the activity of neurons within animals that are moving freely in their environment. For example, we can look at the neurons that produce serotonin and ask how their activity is regulated in the course of normal behaviors. And what Steve did when he found that is that serotonin neurons are actually active in ways that are recognizably related to these long-term behavioral transitions. That when animals make a transition from a roaming to a dwelling state, their serotonergic neurons have a long pulse of activity. It's actually about 2 minutes long that marks that transition. And conversely, the activity of the serotonin neurons falls when the animals move from a dwelling to a roaming state. So this system is not sort of generally permissive. It's actually time correlated to the behavior we're interested in. And that time correlation extends to functional correlation, which we can study using the tools developed originally by Carl Dijsselroth, who will speak in the next session, and others, to directly regulate the activity of these neurons through optogenetics, through the expression of transgenes that let us turn on and off the neurons with light. And what those experiments do when applied to the roaming and dwelling system is to show that if we imitate serotonin by shining light in the patterns where we see the neurons would be active, we can trigger animals to move from a roaming to a dwelling state. And if we antagonize the serotonin system and block its action, we can trigger a transition from the dwelling to the roaming state. So these modulatory systems are not just correlated with, but directly causatively linked to behavioral states. And finally, we see that the function of these systems interacts with each other in ways that show that each of them has independent and important functions. That if we remove both of these systems, what we end up with is animals that can both roam and dwell, but have very unstable behavioral states that will shift very rapidly between roaming and dwelling behavior. And so from this, we conclude that one of the major roles of the modulatory systems is to generate long-lasting or sustained behaviors over periods of time, in this case, minutes. So in conclusion, we think that the wiring diagram of a worm, and very likely the wiring diagram of more complex nervous systems, includes a set of different wiring diagrams, fast synapses that are used for perception, locomotion, and action, and slower neuromodulatory circuits that may create behavioral or circuit states that last for longer times. That the circuits are orthogonal to the fast circuits, that they represent multiple neurons, but still subsets of the neurons. We know that the neuromodulators are multifunctional, that a single modulator carries out multiple functions, and we know that they are instructive. And I would like to point out that, to the extent that we understand these molecules in mammalian systems, many of them can also be interpreted as being important for behavioral state stability and for switching. So for example, hypocretin, orexin, the mammalian neuropeptide, is required for regulation of sleep-waking states, and it's disrupted in the disorder narcolepsy, cataplexy. Dopamine in Parkinson's disease is best understood as being involved in initiation or inhibition of action, of switching between active states. And I'd like to put a plea in here to say that the neuromodulators are really understudied, and they are so strongly implicated in important aspects of motivation and emotion. There aren't even knockouts or conditional knockouts for most of the mammalian neuropeptides. And yet the human brain has many overlapping circuits and many neuropeptides and neuromodulators. And we can think of the wiring diagram as a set of options that neuromodulators help select among those options, and that perhaps by understanding them better, we could also interact with them more productively. And with that, I will thank you very much for the invitation and for your attention, and look forward to the rest of the afternoon.